Dr. Douglas Axe is a molecular biologist and chemical engineer, a Caltech PhD, and was a postdoctoral research scientist at the University of Cambridge in England. He is the director of the Biologic Institute in Redmond, Washington.
Thank you, Steve. I will spare you the 8-hour lecture on gene expression. Instead, we'll do just about 45, 50 minutes on this, which is kind of the big picture story. I spent about 20 years now after Caltech, moved into a postdoc, became very interested in testing whether molecular evolution works or not as advertised, and scratching my head and trying to think about how you would go about determining whether it works or not. And I've come to a very unambiguous conclusion that it does not work. And so myth is in the title for that reason. But what I want to do first of all is talk about the story that I'm claiming doesn't work. And you can decide for yourself whether it works or not. I'm going to give only one, at the end I'll give one experimental example of a way that we've tested it that I think is particularly telling about the failing of the mechanism. And it's one of many that I could give. So molecules to man is this sweeping story of not only Darwinian evolution, which starts with the origin of life, but pre-life, where you have a lifeless planet Earth with water and lots of inorganic small chemicals, but nothing complicated like you have in life. And somehow, through volcanoes or lightning strikes or thermal vents in the ocean, some circumstances came about at some point that gave rise to the first primitive protocell, which developed into a cell as we would recognize it, which is DNA-based, would have had a chromosome, would have been very much like a modern bacterium. And the DNA encodes proteins, and the proteins are the actual workhorses that do the chemistry and make the machines inside the cell. So that's the story for the origin of life. And from there, Darwinian evolution supposedly kicks in, according to this story. Once you have life, you have the possibility of natural selection, and you have the possibility that life will diversify and go in any of a million different directions, which it actually did, according to the story. And we are one of those directions, but we are only one of millions. And the man most associated with the whole story is Charles Darwin, although he did not actually address the first part. He only addressed the evolution once you have life. But because he came up with his theory in 1859 before we knew anything about the molecules of life, he did not speak of DNA. He did not know what a gene was. And it was really up to a number of later workers in the middle 20th century. I've shown here 4 of the key ones: Haldane, Huxley, Fisher, and Wright. These people picked up Darwin's idea in the 1930s and 1940s and framed it around this new understanding of the genetic material, which is DNA, which we now know of as DNA and genes. And so it's kind of Darwin 2.0, modernized it, brought it up to date with Mendelian genetics. None of these guys touched the origin of life issue, though, but several people around the same time, a little bit later, early 1950s, a guy named Stanley Miller, shown here, working in the laboratory of Harold Urey, did a very famous experiment where he had some of the gases that were thought to have been present on the primitive Earth, ammonia, methane, hydrogen, in a glass flask. And he was a graduate student. This is posed much later. He was a graduate student when he did this. Glass flask, had electrical discharge going, and left the thing going overnight. You can imagine his excitement when he arrives early in the lab. First guy in the lab in the morning, opens the lab door to see what happens. And there, he saw Frankenstein. No, it wasn't Frankenstein. It was much less exciting than Frankenstein. It was brown goo in the bottom of his flask. But it was an interesting kind of goo in that it contained amino acids. And amino acids are one of the building blocks of life. So he had successfully transformed inorganic small molecules into organic molecules. A whole lot of excitement over that. But I have to say, in the 60 years since then, if you had asked Miller or many other scientists in 1952, where will this go in 60 years? They would have said, we will have solved the origin of life easily by the year 2000. It just didn't happen. And in fact, it's retreated somewhat because we now, few scientists accept his formulation of what was present on the early Earth as being accurate. And it turns out that it's hugely problematic if you have any oxygen in there at all. You don't get the same result. But Darwinism was born in 1859, long before you could look at molecular mechanisms. Even this rebirth, Darwinism 2.0, was 1930s, 1940s, before the birth of what we would call modern molecular biology, which really started, most people would pinpoint the beginning as 1958. So here's the interesting thing. If Darwin 2.0 is a molecular-based theory, if the mechanism is molecular, and it was the ink was dry on it before molecular biology was born, then what did the evolutionary biologists do at this point when they saw the birth of a new science that could now examine rigorously the basis of their own theory? Well, scientists are all about truth, right? So the evolutionary biologists would have said, okay, we've done our work, we've framed a theory, we had no idea whether it's true or not, but now you, molecular biologists, you can take over now, and you can examine the very mechanism of our theory, and you can tell us whether our theory is correct or whether it's partly wrong or whether it's totally wrong. But we bow now to you. You take over from here. And of course, because scientists are entirely interested in the truth, that is what you would expect to happen. Well, I'm going to take some excerpts that I'll read from a textbook that appeared in 1963, right as this was underway, right here, written by two prominent Stanford biologists, Ehrlich and Holm, and it's about this very mechanism of evolution. They are evolutionary biologists, not molecular biologists, but it's interesting because the book that I'm going to quote from came out right as this was happening, and it's clear that it was happening. The genetic code was in the process of being cracked. Here's their summary of how it all works. Even a slight advantage or disadvantage in a particular genetic change provides a sufficient differential for the operation of natural selection. Thus, the property of light sensitivity of unicellular organisms provides a starting point for the development through selection of the highly complex eyes found in vertebrates, insects, and certain mollusks. I'll translate that into pictures. This is called sensory rhodopsin. It's a protein complex shown here in part because we don't have the full structure for the transducer protein here. So these two lines represent the membrane of a bacterium. And this protein complex sits inside the membrane, and this would be the outside, this would be inside the cell. And this remarkable little complex, molecular scale here, senses light and triggers a cascade of signals that go through the transducer that allow the bacterium to respond with flagellar motors and either swim to the light or away from the light, depending on what the bacterium is trying to accomplish. So this is a light-sensing device, okay? This is also a light-sensing device. This is a compound eye, a rather beautiful compound eye on a soldier fly. And it is true, I'll grant to Ehrlich and Holm that there's at least a conceptual connection between these two things. But will you grant me that this is a whole lot more complicated than this? This is a molecular scale thing. This is a fly eye. You can see this. It's huge. It's made of thousands, tens, hundreds of thousands of cells. Each of these cells has tens of thousands of proteins, each of which is at least as complicated or more complicated. So there's really, although there's this conceptual connection, there's almost no comparison between what we're looking at. And to say that the one explains the other seems to me like an avoidance of science, avoiding an opportunity to do science. Here's how I would say that Ehrlich and Holm, and not just them, but the evolutionary biology community is reasoning. They're using this equation: similarity plus an evolutionary story equals an evolutionary explanation. And if you believe this, then you can give, That's how you can give the details the back of your hand. Poof, who cares? I've got a story and I've got similarity. I don't need the details. You don't need to do the science. That seems like a shame to approach science that way to me, and that's not the way that I've approached it. When someone tells me that I should not look at the details, that's the first thing I want to do is go look at the details. So let me illustrate with something more familiar than molecules for most of you, I assume. What goes wrong with that equation, with similarity plus story? You may recognize these as automobiles. And there is a certain similarity, at least of concept and somewhat of form. We know the work and the intelligence that goes into this, but suppose someone were to propose something that's more Darwinian in nature, that the entire history of automobile invention could be explained without intelligence by this theory. We'll call it the grand story, innovation by manufacturing defect. And it works like this. We all know that cars come off of assembly lines. So you have a factory that cranks these things out, and each model has its own assembly line, of course, because they're very part specific. And occasionally, assembly lines make mistakes, and usually those mistakes are not good, and the customers reject them, and they fix whatever went wrong on the assembly line. But on rare occasion, according to this theory, a mistake actually turned out to be a hit, and the people love it. And so then the owner of the company either retools the assembly line to take this mistake and now make it a feature, or they spin off a new assembly line and keep the old one, so they have two models where you had one before. And according to this theory, you can accept it or you can be skeptical. According to this theory, this is how all of automobile history came about, through manufacturing defect. Well, if we want to view history that way, what we would do is look at the cars that have been produced during these different eras. We don't have the assembly lines anymore, except for the very recent ones, but we can infer things about the assembly lines by looking at the parts of the cars, right? You know what parts were put together if you look at the cars. And since we know that according to this theory, that the assembly lines are related to each other by these mistakes, these accidental mistakes that happened to sell cars, we could propose a history, a specific history that is a working out of the grand theory. And we could say this assembly line came from this assembly line when there was an error in, the Mustangs in 1970. The Beetle came from an earlier Beetle factory. And we could try to apply this story about design error leading to new assembly lines. And of course, because it's not true, because the whole thing is false, there isn't going to be any one answer to this. You could argue this any way you want, and you could get in fights over it, and people could do this until the cows come home. And no one, I guarantee you, no one will propose a history that's absolutely compelling so that everybody says, that's it. That has to be it. Why? Because they're all totally false. That's why. And the other interesting thing about this is if you had a community of people who were convinced otherwise, that this is true, they could go on playing this game of trying to tell the detailed history forever. They could do this forever. If they got a paycheck for doing this, nothing in the exercise of this would force them to stop. Nothing would force them to stop. They would have to step out of this exercise and say, hang on, are we possibly wasting our time here? Otherwise, keep getting your paycheck, keep telling stories, keep writing papers, keep arguing over the details of these trees. Is something like this possibly happening in biology? I would suggest that it is. It's now widely acknowledged that all the tree building in biology has run into huge problems. There's no consistent tree. The Telegraph, British paper, Charles Darwin's Tree of Life is wrong and misleading, claim scientists. In the technical literature, the same thing but in more technical language. Tangled trees, the challenge of inferring species trees. Gene tree discordance. I've got a little excerpt from that, from this paper that says this: many of the first studies to examine the conflicting signal of different genes have found considerable discordance across gene trees. Studies of hominids, pines, cichlids, finches, grasshoppers, and fruit flies have all detected genealogical discordance so widespread that no single tree topology predominates. In other words, it's a mess, okay? Does anyone step outside and say maybe the whole thing is predicated on nonsense? Or do they just say, well, I mean, this is complicated. We need more money so that we can solve it. Well, what's the root of the problem? I claim it is not the detailed histories themselves. In other words, we don't just keep doing this until we get the right one. That's not the root of the problem. Now, you might say that the root of the problem is the grand story, this grand Darwinian narrative. If that's false, if we're making our detailed histories predicated on something that's false, that certainly is a problem. But I don't think it's the root of the problem. This, I think, is the root of the problem. The idea that we are doing science by telling stories. Because if we got rid of that, then someone would do the science and take it seriously and say, oops, we may have made a mistake. Our big narrative, our Darwinian narrative may be totally wrong. That, I think, is the root of the problem. So, instead of just insisting that natural selection is so powerful that because these are both light-sensing devices, this explains this, perhaps we should view that as a question. It's very reasonable for someone to say, I propose that this could evolve into this. But if they do propose that, boy, they're going to raise a whole lot of questions, and I hope they're willing to engage the science, even if it does not bear that hypothesis out. So here is the one experiment that I'll talk about very briefly that I've done with my colleague Ann Gager. We did ask this question, and because we're very skeptical of this kind of transition, we asked it on a much more modest scale. What we did is we looked through the protein databases and all the protein literature, and we searched for two proteins that are as similar as we could find structurally that perform distinct functions. And what I mean by distinct, here are the two proteins we found. One's called Cable, one's called BioF. We'll just call them purple and blue here. You can see that they're very similar. But if you knock the gene that makes purple out, so if you remove that from a bacterium and you give it blue, it cannot do the job. Blue will not substitute for purple, and purple will not substitute for blue. So there's just a very real scientific fact that means these are different. One will not do the role of the other. And yet they are very similar, and they use very similar chemistry. They just perform a different specific reaction. This is a bit abstract, but we reported this all in a paper. And this is a diagram that is taking this whole big supposed family of related proteins. There's about 57, 58 of them. They're called PLP transferase proteins. And what we did is the structures are known for these. We did a structure comparison and mapped out, okay, what is the most structurally like what in this family? And this is the sort of tree you get. So in other words, the longer a line is here, the greater the dissimilarity between that protein and that protein. And don't worry about the names. They're just designations of the structures that identify these different proteins. So you get a mapping like this. The dashed lines in this show that there's a jump in, there's a functional change in going from here to here, from there to there. If it's a solid line, then there is no functional change. And in some cases, we have a gray line here where we're not sure what the function is. Well, in this graph, the two proteins, purple and blue, are right here. They perform different functions, which is why we put a dashed line between them, but they are about as close as you can get and perform two different functions. We don't know of an example, at least in one species, that's closer than this. There could be, but this is the closest we could find. And so our goal was this. It was to turn purple into blue, okay? And to do it with as few changes as we could. So this shows the shape. These proteins actually function as what's called a dimer. Two identical halves come together. And if that looks kind of like a left hand, it looks kind of like this. You have the thumb sticking up. It's like a left hand shake, not like left to right, but if I shake your hand, left hand to left hand, it forms the protein thumbs on top, and you have the crevice where the two hands come together, forms a little cavity, and you see these red molecules that are stuck in the cavity. This is where the actual chemistry happens. It's on the inside where the handshake occurs. Molecules come in, they get converted, and they go out. Now the colors here, gray represents positions where purple, this is going to get confusing. Cable to BioF is what we're trying to do, that conversion. Wherever those two proteins have the same amino acid, I've colored it gray here. Wherever they differ, it's going to be brown, yellow, green, or blue. So you can see a lot of the surface is the same on these two proteins that perform different functions. You also see that these yellow, green, and blue tend to be in the vicinity of where the red thing binds, where the action happens. And so our thinking was where you see red, green, and blue here, sorry, there's not red, yellow, green, and blue, red is these. Where you see those colors, those would be a top priority for changing if you want to convert the function, because they're in the place where all the action is happening. We could probably forget about the brown out here because it's so far away from where the action is happening, where the chemistry is happening. Well, so we did this. We changed all of the green, yellow, and blue in order to convert cable into BioF. And if you put these two halves together, it looks like this. Peeking in here, you can see the red compound, which is where the chemistry is happening. And we're looking in through this tunnel, and you can see that there's yellow. I can't see green here, but there is green on that surface, and blue as you come into it. All of these we changed so that we should get the new function, because we've changed everything that's touching where the chemistry happens, and yet it didn't work. It didn't work. And the reason it wouldn't have to, you might think, well, how could it not work? You just changed everything that's touching the chemistry. The reason is there's subtlety to the shape of these things, and the whole protein is part of what's configuring the shape that does the chemistry. And yes, we got the surface changed, but if you have other things out here that are deforming the surface that's doing the chemistry, then you're not going to get the change. And we found out that you just can't get it with a few changes. Here's the paper that describes the result. The abstract concludes with this, if I can read it. We infer from the mutants examined that successful functional conversion would, in this example, require 7 or more nucleotide substitutions. But evolutionary innovations requiring that many changes would be extraordinarily rare, becoming probable only on timescales much longer than the age of life on Earth. Considering that these two proteins, cable and BioF, are judged to be close homologs, close relatives, by the usual similarity measures, this result and others like it challenge the conventional practice of inferring from similarity alone that transitions to new functions occurred by Darwinian evolution. There it is. Kind of technical, but that's the result. So, and it's published and peer-reviewed. So I claim this. Similarity plus evolutionary story does not equal evolutionary explanation. And I think I've proven that. It does not equal a scientific explanation. Now, what we haven't proven is that in some case, you might actually get that transition to work. I'm not claiming that never works. I'm not claiming that there's no example that's even closer to the ones that we looked at where you could bridge the gap. I'm claiming that you can have remarkable similarity and the gap not be bridgeable by Darwinian evolution. And if that's true, and it is, then everyone needs to back off of these similarity stories and say, hang on, I see there's similarity, chimp, human, I get it, there's similarity, but there's some significant differences here, and you cannot assume that there's an evolutionary transition just because there's similarity. Okay, I want to conclude quickly so that we have time for some questions. Another thing I want to say here is this result, if you think about it, I think it goes beyond a critique of Darwinism and into the possibility of saying something about design itself. Because, as we said, although I agree with those two Stanford evolutionary biologists that there is some similarity between this protein complex and a complex I, it's not actually a material similarity, it's a concept similarity. And engineers, designers are very familiar with concept similarities. And that's exactly the same case here. There's a very clear connection of concept, of idea between this beetle and that beetle. And I actually drive the one that's between the two, the old new beetle. And there's a clear continuity of idea, but not material. You can't pull one of these old beetles out of the scrapyard and beat it up and get a new beetle. It just won't work. It's been totally redesigned. There's no material continuity. Accidents will not get you from one to the other. But there is this continuity in advancement of concept. And that's a design idea. And so we think that this is opening, we're trying with our work not just to bash Darwinism. That becomes too easy after a while. We're trying to flesh out ideas that will take biology in a new direction by taking design seriously. I will end with that. Thank you.
To view this program again and share it with your friends, and to learn more about this series, these conferences, and the intersection of science and faith, please go to www.pensmore.org.